Good afternoon, everybody, and welcome to Brittany's Thesis Defense. Before we start the defense, I'd like to explain the order of business for today. In a minute, I will introduce Brittany, and then she is going to present her work to you all. After she's presented, there will be a period for public questions. And then when the public questions are over, we're going to ask everybody apart from Brittany's committee members to log off so that then Brittany can have a discussion with her committee. After that, Brittany will be sent out for a while we discuss her performance. And then we will let Brittany back in, let her know how she's done, and then she will be free to tell all of you what the result is. So that's going to be how we're going to run things today. And during the public question component, please type your questions into the chat, and Brittany will read those questions and respond to them. So thank you again for coming. It's my great pleasure this afternoon to introduce Brittany Evans for her master's thesis defense. Brittany originally hails from the great state of Georgia. Before she came to the University of Hawaii, she graduated from Eckerd College in Florida, where she got a stellar GPA and was awarded a number of prestigious scholarships. Those included a Noah Holling Scholarship and a Barry Goldwater Scholarship in maths and science. She also gained fairly broad research experience before she came to Hawaii. So she did a population genetic study on dusky pipefish. She studied the influence of salinity on oil pollution tolerance of invertebrates. And she also participated in a dolphin photo identification study. Now, in 2017, when I was combing through several hundred prospective students who contacted me to come and work with our group, I immediately picked Brittany out as an exceptional student. And I ended up actually having to compete with other labs, including one of my former graduate student lab mates, to recruit her to the University of Hawaii. And we're very glad that she came. And after she got here, she learned some new skills, including how to drive boats, how to splice ropes, and how to capture and tag enormous sharks. And she was hoping to study deep sea sharks for her master's thesis. But then along came the COVID-19 pandemic, and essentially that became impossible. But fortunately, Brittany was able to pivot to another really impressive marine fish, which she's going to tell you about during her defense. So I'm now going to turn the proceedings over to Brittany. Thank you very much. There you go, Brittany.
All right. Thanks, Carl. Give me 1 second to pull up my presentation, everyone. Let's see. And let it load. All right. Well, like I said, thanks, Carl, for the introduction, and thank you all for coming to my defense today. Before I begin, I would like to point out one thing. I used some Hawaiian words in my presentation, and I tried my best to do justice to these words in respect for the Hawaiian culture, but please forgive me if I don't get them quite right. So my defense was on tracking Ulua movements throughout the Hawaiian Archipelago. And though there's been some differences in technique, the idea of animal tracking has played a long and important role across human history. The movements of animals has been of interest to humans since our earliest days as hunter-gatherers, where understanding animal patterns was crucial to finding food, whether it be trying to find out what bison seasonal aggregations were, or just the simple act of physically tracking deer and bird prints along the ground. Humans have used tracking to learn and predict animal locations throughout human history. In more recent decades, the field of movement ecology has sought to understand the patterns and drivers of animal movements. Advancements in technology have helped us to elucidate movement patterns through the use of telemetry, where scientists put animal-borne tags and use those to remotely record and map an individual's movements through its habitat. For aquatic species, quantifying animal movements can present some challenges, as it's a more concealing environment where these animals reside. For example, satellite and GPS tags traditionally require some level of communication between tags and overhead satellites in order to estimate an animal's location. However, transmissions between the two can be difficult in aquatic environments, often requiring the tag to come out of the water in order to communicate. To overcome this barrier, movement studies on aquatic species often use acoustic telemetry in order to track individuals, as sound travels remarkably well underwater. Acoustic telemetry requires two components: a tag attached to an animal that's periodically sending out a unique series of pings, and a receiver that's continually listening for the presence of these pings. Think of an acoustic tag as a Morse code license plate, periodically sending out its information as an animal passes by a receiver. Once that animal comes within range of a listening receiver, the receiver picks up that transmission and records the animal's unique ID code, date, and time of transmission. This allows two opportunities for gathering movement information. The first is through active tracking, where you go out to the field and manually follow an individual as it moves throughout its habitat. The other is through passive monitoring, where you have a stationary array of receivers moored in a given area that are continually listening for the presence of any individuals that are tagged. Using this stationary array of receivers, you can actually track an individual's movements by looking at which receivers were able to pick up that individual's transmissions. Both acoustic techniques, active tracking and passive monitoring, have their advantages, and together can help elucidate fine-scale movement patterns within a given environment. But why do scientists even track animals? Well, animal tracking allows us to uncover key information regarding movement ecology, answering questions such as, do these animals have a tendency to return to the same areas? And what areas are these animals commonly using? Spatial information such as this helps us make inferences into an animal's biology and ecology. In addition to contributing to the general understanding of a species' spatial ecology, descriptors of movements aid in fishery stock assessments and spatial management of protected areas, as how an individual distributes its time in a habitat has important implications on the management and conservation efforts. Active tracking provides the link between conservation policy and animal ecology, as in order for an area to protect a certain species, it's important to know if the managed area encompasses the animal's commonly used areas. As for marine protected areas, the Hawaiian island chain is home to one of the world's largest, the Papahanaumokuakea Marine National Monument. This chain of largely unfished, uninhabited islands encompasses approximately 1.5 million square kilometers of protected area and is home to a number of threatened and endangered species, tropical reef fish species, and several species of sharks. The monument's protected status and remote location make it largely isolated from anthropogenic impacts, and its coral reefs are regarded as near pristine habitat. This differs slightly from coral reefs in heavily populated areas of the main Hawaiian Islands. Perhaps due to this difference, the abundance and size of top predators in the monument are significantly greater than the predator assemblages found in fished islands in the main Hawaiian Islands. Within the monument, there is one species that dominates the predator assemblages in the coral reefs, accounting for up to 70% of the total predator biomass and almost a third of the total fish biomass. This is the giant Trevally, a large predatory fish in coral reef ecosystems found throughout the Hawaiian Archipelago and across the Indo-Pacific. These fish play an important ecological role as mesopredators in coral reef ecosystems, preying upon a variety of species, including fish, some seabirds, and the occasional sea turtle hatchlings. These predators can reach over 5 feet in length and weigh close to 200 pounds, making them highly prized game fish as anglers from all around the world come to try and hook these fish. In addition, giant trevally are an important species in Hawaiian culture. Known as Ulua aukea, and I'll just refer to them as Ulua from here on, these fish feature in oral traditions and were heavily regarded in traditional practices, where in some sacrificial ceremonies, if an Ulua couldn't be found for the ceremony, a human was used in place as the substitute sacrifice. But despite their cultural significance and likely importance in coral reef ecosystems, we still lack key basic knowledge on Ulua space use, especially here in the Hawaiian Archipelago. Now, there have been other investigations into Ulua movements, including here in the Hawaiian Islands. These studies together suggest that Ulua are site attached to specific areas, and that these commonly used areas are generally less than 10 kilometers. However, there still exist knowledge gaps on Ulua movement patterns, as we don't have information on Ulua from an archipelago scale or over a long time period. For example, are these Ulua traveling up and down the archipelago? Are Ulua that are found in the protected monument coming down to the main Hawaiian Islands where they can be fished? To answer these questions and more, we needed a larger, multi-year, multi-location analysis in order to more fully characterize Ulua spatial dynamics across the Hawaiian Archipelago. And that is where my thesis comes in. My thesis was designed to fill in some of these knowledge gaps by using a long-term acoustic monitoring dataset that was supplemented with some smaller tracking techniques in order to more fully characterize Ulua movements and space requirements across the Hawaiian island chain. My thesis currently represents the largest acoustic telemetry study of Ulua, including the longest total duration of acoustic monitoring, the longest time an individual Ulua was acoustically monitored, the largest sample size of tagged and tracked Ulua, and the largest total monitoring area for any Ulua acoustic telemetry study currently published. But in order to track Ulua movements, we needed to catch these fish. Ulua were captured using hand lines and fishing poles and then implanted with small acoustic transmitters. Here you can see some of the crew fishing for these, mind you, highly prized, highly regarded fish, including my own advisor, Carl, who's captured some fairly large individuals that I think would make some fishermen and anglers quite jealous. In my thesis, 166 ulua were captured and tagged at 5 unfished islands in the monument and 2 fished islands in the main Hawaiian Islands. Here you can see a small view of the Hawaiian Archipelago with labels for the islands where ulua were captured. Ulua were fitted with transmitters all the way from the northwestern portion at Kuri Atoll, all the way down to the southernmost island at Hawaii Island, or the Big Island. As soon as these ulua were released, they began sending out these pings that could be potentially picked up by the moored receivers throughout the Hawaiian Island chain, from anywhere from 2 to 10 years. Now, ulua are found to live up to about 25 years. So some of these ulua could potentially be tracked across the Hawaiian Archipelago for up to 1/3 of their total lifespan. With these ulua swimming around and emitting pings, we needed something to pick up those transmissions. A total of 83 receivers were stationed at 7 islands in the monument and 1 island in the main Hawaiian Islands. Overall, the receiver array's monitoring period overlapped with active ulua transmitters for a total of roughly 14 years, monitoring from 2005 all the way to 2019. This passive acoustic monitoring database was also supplemented with active tracking data, where 3 ulua were captured in waters off of Big Island, put with acoustic pingers, released, and then manually followed by boat for several hours. Though each individual was only tracked for a maximum of 50 hours each, this information allows me to examine fine-scale movement data that's independent of the acoustic array found throughout the Big Island. For aquatic animals, both horizontal and vertical movements are important for understanding how an individual moves throughout the water column. To elucidate ulua vertical movements in the Hawaiian Archipelago, a subset of those acoustic transmitters that we had mentioned earlier were also equipped with depth sensors. Here you can see the tagging locations of the 2 islands where the ulua were tagged with these depth sensors and acoustic transmitters at French Frigate Shoals and Pearl and Hermes Atoll. Each time one of these 41 tags came within range of a moored receiver, that receiver also picked up the tag's pressure reading, which could later be converted into the ulua's swimming depth. To complement this information, those 3 actively tracked ulua in waters off of Big Islands also gave depth readings with each ping. In the monument at French Frigate Shoals and Pearl and Hermes Atoll, ulua were detected at depths ranging from the surface to 80 meters, but stayed generally between 5 to 25 meters. While this suggests that ulua display a strong fidelity to a relatively narrow and shallow depth range, these depth readings could be influenced by the acoustic receiver locations, as an ulua couldn't be detected in waters deeper than the bathymetry limits within the monitoring range. In this case, it's also important to look at ulua depth distributions in relation to the surrounding bathymetry around the receivers. Here you can see those receivers at French Frigate Shoals and Pearl and Hermes Atoll that had depth detections, as well as a white circle showing the maximum potential listening range for each receiver, which was at about 1 kilometer. On the other side, you see those same acoustic receivers along with the depth distributions of the local bathymetry surrounding the receivers in red, and the Ulua swimming depth distributions in blue. Now, the receivers' monitoring ranges were mostly in shallower waters than 100 meters, with occasional insights into deeper waters at around 300 meters or greater. However, Ulua, shown there in the blue, typically stayed within shallower waters than the limits provided by the local bathymetry, suggesting even when Ulua had access to these deeper waters within the detection footprint of receivers, they were rarely venturing into these deeper zones and instead staying within that relatively narrow, shallow depth band. While bathymetric compression certainly can have an influence on Ulua depth readings, the local bathymetry surrounding the receivers alone does not fully explain the depth distribution of these Ulua within the monument. To remove the influences of acoustic receiver deployment depths from our consideration, there was one Ulua that was tagged with a satellite tag in French frigate shoals for several months. This tag recorded the Ulua swimming depth independent of any surrounding acoustic receivers. And the tag shows a similar story, with the Ulua spending most of its time in shallow waters under less than 25, with the occasional short dive to around 80 meters, shown there in that spike at the end. Now, the story of Ulua vertical movements changes slightly when we look at the 3 Ulua that were actively tracked on waters off of Big Island. Here you can see the depth distributions of these Ulua following the hours they were tracked. You see Ulua 1 as A, Ulua 2 as B, and Ulua 3 as the bottom 2 panels C and D, since this Ulua was tracked on 2 separate tracking occasions about a month apart. In general, these Ulua were spending most of their time between 50 and 100 meters, with occasional short spikes to the surface and a maximum dive of 242 meters. Of course, Ulua 1 did show slightly different depth distributions than the other 2, as its distribution looked more similar to those Ulua in the monument, as Ulua 1 was spending most of its time between 10 and 20 meters. However, this was also the Ulua that was tracked for the shortest period of time, so we have less information about its depth readings than the other 2. Ulua active tracking depths off of the Big Island were generally deeper than the passive acoustic monitoring depths found in the monument. Here you can see histograms of the local bathymetry surrounding the tracked Ulua in gray bars, with French frigate shoals and Pearl and Hermes Atoll along the top, and the waters off of Big Island along the bottom. Superimposed on those bathymetry readings are the minimum, mean, and maximum swimming depths of Ulua found in those 2 regions. As you can see, Ulua mean swimming depth in the Big Island more closely resembled the maximum swimming depth at the monument. This Ulua swimming depression may be due to the deeper waters around Big Island, as bathymetry estimates were generally deeper than those in the monument. However, Ulua did have access to both shallow and deep waters in the 2 regions, so it's possible that the difference in swimming depths may not solely be due to the bathymetry readings alone. Instead, it's possible that it could be due to differences in environmental parameters like water temperature. Or it could be due to the heavier fishing pressure in the main Hawaiian Islands, as more shallow dwelling fish maybe are more likely to get fished out. However, to say more on this topic, More studies into Ulua depth distributions are needed to precisely describe the variation in Ulua depths between the different islands, as well as examine the factors that could be influencing those variations. Although it's important to examine their vertical movements, Ulua horizontal movement patterns are also a crucial component in understanding how an animal uses its habitat. The term site fidelity is a common metric used to describe an animal's horizontal movement patterns in the scientific literature. Site fidelity refers to the tendency of an individual to reside in a given area and is often measured by, well, its Site Fidelity Index, or SFI for short. In this thesis, SFI was calculated as the number of days an Ulua was detected divided by the number of days the Ulua was monitored, times 100 to make it an easier percentage. In my thesis, SFI and 3 other detection metrics were used to evaluate site fidelity, as each detection metric tells a slightly different story, but collectively provides an overview of how intensely Ulua were using these locations monitored by the receivers. Although I found evidence of Ulua site fidelity to core areas, SFI values varied between individuals, with some Ulua exhibiting high site fidelity and others showing consistently low SFI values of less than 1%, meaning for all the time that these receivers were listening for that Ulua, the Ulua was only detected on about 1% of those days. These low SFI values of some individuals could be an artifact of sparse receiver coverage, as it's possible that these individuals did have high site fidelity to a given area, but that area was just outside the monitoring range of my receivers, so I couldn't detect it. Of course, these low SFI values could also mean that these Ulua just moved more extensively than others, and perhaps didn't show fidelities to a given area. Overall, the SFI values show us that Ulua can have moderate to high site fidelity with some variation between individuals. Site fidelity is a commonly observed behavior in animals, suggesting there's some evolutionary benefit to repeated use of a spatial area. An animal's familiarity with an area could help it more effectively acquire resources, catch prey, or avoid predators in certain environments. This tendency towards stronger site fidelity has been seen in other Ulua studies and suggests that most Ulua have well-defined core areas. These core areas are what's known as home ranges. Like site fidelity, home range is another common term used to describe horizontal movement patterns. Though the exact definition has been of considerable debate, I'm defining a home range as the core area used on a repeated daily basis by an Ulua. If Ulua have well-defined core areas and well-defined home ranges, they should produce pretty characteristic patterns of spatial detections. For example, take this dotted circle to represent an Ulua's home range, and the stars to be the latitude and longitude positions of various acoustic receivers. Detection should be more frequent within that home range boundary, shown as sort of the gray color, and become less frequent as you move away from those home ranges, shown in this gradient to white. We can look for this pattern using acoustic telemetry by seeing if the number of detections on a receiver start to decrease as the receivers move away from the Ulua's home range center. However, we don't actually know what the Ulua's home range center is. Instead, I assumed each Ulua has the highest probability of being captured within the core area of their home range, as this is where they should be spending most of their time. Therefore, I used each Ulua's capture location as a proxy for home range center. Now, I checked my assumption on this. If Ulua had well-defined home ranges and capture location served as an adequate proxy for home range center, we should see a fairly strong negative correlation, as what's shown here in this idealized line graph, where the number of detections on the y-axis start to decrease with increasing distance away from the tagging site, until these detections start to level out once you hit past the home range boundary, shown there in the dotted line. And that is exactly what we see. Here you can see the 4 scatter plots showing the 4 different detection metric values along the y-axis, including SFI that we talked about earlier in A. Along the x-axis is increasing distance away from the Ulua's tagging site. In all 4 of these scatter plots, detection metric values decreased as you moved further away from the tagging site. Statistical analysis through multivariate adaptive regression splines identified the inflection point at which the slope, this decline in detections, started to taper off. This leveling off point, shown as the red lines in each of these scatter plots, is what we can assume is the general boundary of the home ranges for these Ulua in order to estimate their overall size. In each case, that boundary was at between 4 to 5 kilometers for all of these detection metrics, which is suggesting that Ulua have roughly 4 to 5 kilometers sized home ranges. Active tracking data also supports this idea. Here you can see the actively tracked paths for all 3 of the Ulua that were tagged with those acoustic pingers in waters off the Big Island. Now, Ulua 1 over at the top was the one that was only detected for around 10 hours in comparison to the others 40 to 50 each, so it has a much smaller track. But Ulua 2 and the 2 tracking sessions of Ulua 3 show that the maximum linear dimensions for the tracks were between roughly 4 to 5 kilometers, matching our estimates from the passive acoustic monitoring detections. This Ulua linear home range estimate of 4 to 5 kilometers is also consistent with other Ulua acoustic telemetry and mark and recapture studies, where the average linear distances were less than 10 kilometers. While this shows Ulua have relatively small home ranges, we still need to know about the total extent of their movements throughout the Hawaiian Archipelago. Are these Ulua leaving their home ranges for short periods of time to travel to different islands? Well, though there were acoustic receivers deployed throughout the island chain, listening for these Ulua for several years, no Ulua was ever detected moving between the monitored islands. Instead, Ulua had maximum linear distances of roughly 87 kilometers. What this suggests is that though Ulua have home ranges of around 4 to 5 kilometers, they are briefly making much longer trips to around 90 kilometers, but still staying within the island they were captured at. This does coincide with what has been seen in previous mark and recapture studies in the Hawaiian Islands, where ulua were always recaptured less than 80 kilometers away from that initial tagging site. In addition, other ulua studies show that ulua have limited inter-reef movements and relatively restricted movements within their core areas. Deep ocean channels and the distances between coral reefs on adjacent islands may serve as geophysical barriers for ulua movements. Of course, it is also possible that ulua are traveling to different islands, but that these movements were outside the detection range of my acoustic receivers. So the main conclusions from ulua horizontal movement patterns in my thesis were that ulua had moderate to high site fidelity, had small home ranges, and exhibited no inter-island travel. This behavior suggests that ulua are able to meet their routine resource needs within a relatively confined area. Though ulua are large mesopredators capable of leaping up and swallowing seabirds, their movement patterns are most similar to those of smaller reef fishes, which show a strong site attachment and relatively restricted movements to a single coral head or a certain reef, albeit for an ulua on a much larger scale. The conclusions of this thesis have important implications toward the management of ulua fisheries and marine protected areas. Higher site fidelity and smaller home ranges suggest that ulua are resident to given areas rather than transient roving visitors moving from island to island. Therefore, care must be taken to avoid overexploitation of these reef mesopredators. The elimination of fishing has certainly protected the ulua in the monument, and clearly one of the largest MPAs in the world is big enough to encompass an ulua's home range of roughly 4 to 5 kilometers. However, it's possible that other MPAs that have ulua present may be too small to fully encompass an ulua's home range within their protected area. But to say more regarding this idea, we need more studies on ulua home ranges throughout the Indo-Pacific in order to more precisely evaluate the unique effectiveness of each individual MPA. Even here, we need more studies to elucidate fine-scale movement patterns of ulua in the main Hawaiian Islands in order to better determine more effective management strategies. For example, Kealakekua Bay Marine Life Conservation District off the waters of Big Island has certain fishing restrictions within its boundaries, shown here in the blue. While this may be useful to certain reef fishes, judging solely on those 3 active track paths of the ulua near that area, the MPA would not provide enough protection for ulua's commonly used areas, nor would it fully encompass an ulua's home range should an ulua reside in that area. But remember, my thesis only had these 3 individuals tracked for several hours. So for us to say more about the protection of the bay, we need studies on ulua movements both within the bay and slightly outside of it in order to say more about the effectiveness of that particular district. In addition to these questions on ulua movements that need further studies, I will also be working on more analysis for my thesis. Future directions for my research include looking at temporal effects on movement patterns in ulua in the Hawaiian Archipelago. For example, ulua have been suspected to spawn under summer full moons. And it's been suggested from other studies that ulua movements might shift slightly during these spawning conditions. In the Hawaiian island chain, French frigate shoals has been identified as a potential spawning location. So I will be looking at the temporal patterns of detections from the acoustic receiver array and trying to see if I can identify other potential spawning locations. In addition, I'll be looking into temporal effects on day versus nighttime movements, or how movements might change over seasons or over years as the Ulua grow older. I've already found some temporal patterns in Ulua vertical movements within the monument, where there seems to be a seasonal variation on Ulua average swimming depths. But certainly more temporal analysis is needed for us to uncover more information about this. In conclusion, my thesis covered the largest acoustic telemetry study of Ulua to date, monitoring 166 Ulua over 14 years across the Hawaiian Archipelago. Though there was evidence for more narrow depth ranges for an individual, as a species, Ulua depths ranged a couple hundred meters. As for horizontal movements, there was no inter-island travel recorded for Ulua in the Hawaiian island chain, as Ulua were only detected on the island they were captured at. Ulua were commonly site attached to relatively small home ranges, which could have important implications for their ecology and management and conservation efforts. More information is needed to elucidate fine-scale movements of Ulua in the main Hawaiian Islands and other parts of the Indo-Pacific. And I personally will continue to work on this data set, examining possible temporal patterns and potential Ulua spawning grounds. This thesis was built off a very large database. And of course, with a database such as this, it requires a lot of manpower. For all of those people who have assisted with the collection of this database over the years, thank you. Thank you for working on this and allowing me to pull a thesis out of your work. Of course, it would also not be made possible without the funding sources for the collection of this data. And of course, this thesis wouldn't at all be possible without my advisor, Carl. I'm sure a lot of you guys know Carl, but as his grad student, it might be a little bit biased, but he's one of the best advisors. I came to these programs looking for a great advisor, and that's what I found. Carl is an amazing scientist, an amazing mentor, and is willing to be involved in fun situations and get down to serious business when we need it. So I say to Carl, thank you for everything you've done. This wouldn't have been possible without him. And of course, to my amazing committee members, as Carl was talking about, I had to switch my thesis over in the middle of it. And both of these gentlemen have provided so much support and guidance and encouragement. Truly thank them for their help with this thesis. And of course, the Shark Lab themselves, some amazing individuals who have shown me so many opportunities and have helped in my graduate program. And though I think I'm the only graduate research right now that's not dealing with sharks, I think my Ulua have a good place in the Shark Lab. They have a pretty good rep. Lastly, I would like to thank my friends and my family. Without them, none of this would have ever been possible. So thank you. And with that,